Hi, science friends. Another day, another lesson in gene expression. So, today, what I want to talk about is differential gene expression. And so, what that means is how do we have lots of different cells that contain thousands of genes, but they don't all express the same genes? Okay, every cell kind of expresses its own unique subset. Out of those thousands of genes, an average cell is only going to express about 20 of those genes, just because of what it does and the genes that it needs for its job. Because remember, cells are specialized. Some cells are even more specialized than other cells, and so maybe they express even fewer. And so, this idea of expressing different genes and having the same genome but expressing different groups of those genes is referred to as differential gene expression. And so, gene expression, as we mentioned yesterday with the operon, generally is discussing things that happen in transcription. But gene expression can actually happen at lots of levels. So, here in this diagram, this is a eukaryotic cell. We know that because we have the nucleus here. But there's lots of steps. And so each of these steps that I'm kind of highlighting here represents an opportunity for gene expression to be controlled or gene expression to be regulated. Okay? Now, in prokaryotes, we don't have this many steps. We've talked about that prokaryotes don't have that shuttle from the nucleus to the cytoplasm. They don't have that RNA processing, and that their process happens a lot more quickly, almost simultaneously. And so for that reason, prokaryotes don't have the same level of differential gene expression that eukaryotes do. Okay? So one way that we see differential gene expression is in something that's called epigenetic inheritance. And so tomorrow you are going to watch a video. It's a little bit older, but I think it's really interesting. So I know, Fridays are hard to kind of motivate yourself to do some work when you're home. But this video, every class that I've ever shown it to is really interested in it. And so it shows epigenetics. And so epigenetics is interesting because we see traits that are transmitted by things that are not directly involved in the nucleotide sequence. Okay? So, for example, twins are a big part of how we study epigenetics. So, twins have the exact same set of DNA. And so, naturally, we would think that they should express all of the same genes. But that's not necessarily what we see. Sometimes we will see that one twin in an identical twin set will acquire a genetically based disease, but the other one is normal. For example, schizophrenia. So what causes that? Why do we see that difference? And so some examples of epigenetic inheritance are what we're going to talk about in just a second: histone acetylation and DNA methylation. And so those are just some modifications that happen to the DNA that cause the genes, even though they're the same and even though they have the same code, to be different and to be expressed differently. And so that's epigenetic inheritance. Traits that are transmitted by things not directly involving the codes that are read in the amino acid. Okay? How two people can have the same amino acid sequence but inherit things differently because it's something outside of that amino acid sequence. So that's what epigenetic inheritance looks like. So let's talk about these two examples here: histone acetylation and DNA methylation. So histone acetylation. Remember, histones are these purple proteins here that have the little tail on them, and that's what chromatin or DNA coils around to condense and form chromosomes, right? We see this when the chromosomes pack together in prophase. during mitosis and during meiosis. It condenses to form chromosomes, and it's being wound up around these proteins called histones. Okay? Now, this picture right here is unacetylated histones. So they don't have that acetyl group attached. Versus these over here have the acetyl groups attached, which that's just these little green dots right here. That's our acetyl groups. And what the acetyl groups do is they neutralize the charge in the histones. And so basically they don't bind up as tightly. They don't, form this mass here. And so as a result, the chromatin forms a looser structure. We see this loose structured chromatin over here. That loose structured chromatin is referred to as euchromatin. If chromatin looks like this, it's called euchromatin. That's like U means true, so that's true chromatin. Remember, chromatin looks kind of all stringy and strung out, and so that's how I remember that one. This one over here is referred to as heterochromatin. When those histones are unacetylated, it causes it to wind up really tight, okay? And again, that's the DNA, that blue part right there, wound around those histone proteins. Well, which one of those, euchromatin or heterochromatin, do you think is better for transcription? Which chromatin type is going to be more easily transcribed? Okay. If you said euchromatin, you are correct. More easily transcribed. Okay. What we see in heterochromatin is that highly condensed chromatin, or this heterochromatin, is not usually expressed. Polymerase has a hard time getting in there and binding to that, okay? Because of the way that these histones are acetylated. It has nothing to do with the A's and T's and G's and C's, but it has to do with these acetyl groups, okay? And so that's important. That's one way that we can regulate gene expression, all right? The process of removing those acetyl groups is called deacetylation, all right? And they can be removed by enzymes and modify that structure, okay? Now, another thing similar to acetylation is DNA methylation. So we talked about methyls in biochemistry when we did our functional group. A methyl group is a CH3. That's what the functional group methyl looks like, okay? And so methyl groups are attached to bases, usually cytosine, in the DNA, in DNA methylation. And so what we see here with DNA methylation, inactive DNA is typically more methylated than regions that are actively transcribed, okay? So for expression, we want our DNA to be demethylated and we want our DNA to be acetylated. If we want those genes to be expressed, then it's going to be, those genes would be turned on, okay? If we're trying to turn off those genes, we want methylation and deacetylation. And this is the euchromatin. And this is the heterochromatin. Okay? And so those are, again, just changes to the DNA that affect the gene expression. Another thing that affects the gene expression is the presence of control elements. Okay? And so what control elements are is they're non-coding DNA that serves as the binding site for transcription factors. Remember, the transcription factors are what allow polymerase to bind. But what allows those transcription factors to bind is the control elements. So if the control elements aren't there, then the transcription factors aren't going to bind and polymerase is not going to bind. If the control elements are there, then you're more likely going to have transcription. So if we increase the control elements, we increase the likelihood of transcription of those genes. Okay? So these control elements can be proximal. The term proximal means near, and so what we're talking about is near the promoters, or distal. Distal means far away. If you've taken any of the medical classes, either through PLTUW with me or with Miss Neal or Miss Lawrence, you're probably familiar with those terms, proximal and distal. But the distal ones are going to be further away from the promoter region. And then they're also referred to as enhancers, okay, because they are going to enhance gene expression. So here we have some proximal control elements that are located closer to that promoter versus down here the distal control elements located more upstream and further away from that promoter, okay? And so when those are present, then these activators will bind, and that causes these transcription factors to be able to bind right here, okay? And that allows for polymerase to bind and start the initiation or the transcription process, okay? So take a look here at this example. What we see is that activators for a specific protein are only present in the cells that need that protein. So we've got two different genes up here that we're looking at. Albumin is a gene that's involved, it's a blood protein, okay? And then crystalline is a protein that's found in the eye, okay? And so the eye is not going to need to make the blood protein and vice versa. But the genome that, in both of those cells, whether in the blood or in the eye, contains the genes to be able to make both of those. So why is albumin not made in the eye and crystalline not made in the blood? Well, it has to do with these control elements, okay? And it has to do with these activators. So the activators so here's a liver cell, okay? So that's going to be relating to the blood. And then here's the lens cell. So that one's going to be relating to the eye. So the activators that are present in the lens cell, the orange, the gray, and the pink, are going to cause the synthesis of the crystalline gene because those activators are present. And so it's going to allow the transcription factors to bind and the control elements to bind and everything to proceed, okay? Versus in the blood or in the liver cell, the red, the gray, and the yellow activators are present. And so we see those here. Those control elements are present. And so they will, in that cell, cause that expression of that gene. And so we see that the albumin gene is expressed in the liver cell, but the crystalline gene is not because those activators aren't there. And vice versa over here. The crystalline gene is expressed because those activators are there, but the albumin gene is not. Okay? So again, that's another example of how we can control transcription within the cell. Okay? And so the transcription factors that are made in each of those cells determines which genes will be expressed there. All right? Now, let's take a look at one last thing. We talked yesterday about the operators and the operons in the prokaryotic cell, but we said that eukaryotic cells are not like that. Now, this is not fully understood, but when we look at chromatin, which contains all of the DNA in the nucleus, each of these colors here represents a different chromosome. So maybe purple is chromosome 1, light blue is chromosome 2, red is chromosome 3, so on and so forth. But what they do is these distinct chromosome regions, when you look at them up close and you examine them, they kind of overlap with one another and they form these loops. And so it is thought to be that these overlapping regions are kind of a transcription factory that synthesizes genes or proteins for a common function. Just like in the tryptophan example yesterday. So in the operon, that operator controlled all 5 of those genes that was involved in the tryptophan pathway. Okay? And so maybe this is a similar scenario as for how that's done in eukaryotes. We know they're not controlled with an operator in eukaryotes. And again, this is not fully understood, but that's what's believed to be happening here in this overlapping region. All right? So, this lesson is probably less of a tested concept for AP, but what I really want you to see is the big idea that there are things outside of just your A's and T's and G's and C's that control us, right? There are these other things, the way that DNA is modified, whether or not polymerase can activate or can access the DNA. Lots of other different factors that are involved in determining whether gene expression happens, okay? So that's kind of the bottom line and the takeaway message for today. All right, I hope you all have a great day, and I will see you soon.